To unpack this dense research paper, I'm going to provide a little bit of information about Claudin 18.2 and gastric cancer. Gastric cancer, also known as stomach cancer, is one of the most common and deadly cancers worldwide. And despite many advancements in treatment, survival rates for many patients are still disappointingly low. And this has led researchers to search for brand new pathways for treatment. The research study we are covering today focuses on one route that involves the connection between a promising new target protein called Claudin 18.2 and the tumor microenvironment of gastric cancers. Claudin 18.2 is a protein that is part of tight junctions that are found mainly on the surface of stomach cancer cells, which makes them a prime target for therapies designed to attack cancer cells while sparing the healthy tissue surrounding them. However, not all patients with high levels of Claudin 18.2 respond well to these treatments. This suggests that there's a lot more going on inside the tumor environment beyond just the cancer cells. This led the researchers to focus on a well-known factor of growth and migration of tumors known as cancer-associated fibroblasts, or CAFs. CAFs are a small part of these complex tumor microenvironments, and they help cancerous cells migrate to healthy tissues and protect the cancer from the immune system, making it harder for the cancer to be eliminated. Because of this, the researchers became more interested in CAFs as a therapeutic target along with the Claudin 18.2. This study aimed to explore how Claudin 18.2 affects the way cancer cells physically interact with these fibroblasts, and specifically how the interaction might help these tumors grow faster and spread more easily. They hypothesized that Claudin 18.2 might be directly facilitating contact with fibroblasts in a way that fuels tumor progression. And by understanding this relationship, the researchers hope to uncover why some tumors resist Claudin 18.2 targeted therapies and whether there's a need to develop combination strategies that also target the surrounding fibroblasts. To investigate this connection between Claudin 18.2 and CAFs, the researchers used a mix of real patient data and lab-based experiments. They analyzed tumor samples from 185 patients with gastric cancer, and these cells were preserved using a method called formalin-fixed paraffin embedded, or FFPE. This process keeps the tissue and proteins intact over time so that researchers can analyze them. To detect how much Claudin 18.2 was present in these tumors, they used a technique called immunohistochemistry, which involves staining the tissue so that they can visually identify and measure the protein. This allowed them to compare Claudin 18.2 levels and different levels of aggressiveness in tumors. Along with patient samples, they worked with gastric cancer cell lines and fibroblasts that were isolated from human tumor tissues. By growing these two types of cells together in what's called a co-culture, they could observe how cancer cells and fibroblasts behave when placed in the same environment. It's not a perfect replica of what happened in the body, but it's a very useful model for studying interaction between these cells. They used a common technique called Western blotting to measure the amount of Claudin 18.2 in the cells. And they also used a technique called RT-qPCR to look at the levels of different gene expressions when Claudin 18.2 was present. They used a technique called siRNA, which is a tool that can be used to temporarily silence specific genes, and they used it to reduce Claudin 18.2 levels in cells. The absence of Claudin allowed for the researchers to gather more insight into its function with these fibroblasts. They performed adhesion assays, which could measure how strongly cancer cells stuck to fibroblasts and how they aided in migration and in invasion. To test how they behaved in a living system, the researchers used mice as models. To do this, they labeled gastric cancer cells with a glowing green protein called GFP so that they could track them. Then they injected these cells into the mice with and without fibroblasts. This allowed them to watch how the cancer behaved in a live animal. The researchers also used a tool called TurboID, which is a relatively new technique, which allowed them to identify which proteins were physically interacting with Claudin 18.2 inside the cancer cells. Using this array of methods, the researchers could identify how Claudin 18.2 interacts with CAFs.
Going over the results of this study, there were several key findings. The first of which is that patients with more CLDN 18.2 typically had more aggressive stages of cancer. Looking at the figure, an H-score is a measure of how much CLDN 18.2 there was in a patient, with higher H-scores corresponding to higher presence of CLDN 18.2. When those higher H-scores were seen, higher stages of cancer or more aggressive forms of cancer were associated with that, and conversely, lower H-scores were associated with less aggressive stage 1 and stage 2 cancers. Additionally, it was found that CLDN 18.2 attracts cancer-associated fibroblasts, which work in tandem with the CLDN18.2 to make the cancer more aggressive. This was found by looking at indicator proteins such as FAP, alpha SMA, and S100A4. Those specific proteins signal to the researchers that there are CAFs present. These figures at the bottom, which show an upward linear trend, indicate that the more CLDN 18.2 there is, the more indicator proteins there are, and likely the more CAFs there are, which lead to a more aggressive cancer. Next, the experimenters looked at how CLDN 18.2 and CAFs move and traverse within the body. It was found that CAFs can really increase the amount that the cancer can move. In the last two columns, you can see a lot more glowing green. That's because a construct called GFP was attached to the cancer cells to show their movement. However, you can see that the most aggressive cancer, or the cancer that spread the most and therefore showed up the most glowing, was that which had both CLDN 18.2 and CAFs, which is the last column right here. And that really shows how the cancer is most aggressive when those two are together. In live mice, those combinations of CLDN 18.2 positive cancer and CAFs form really dangerous clusters that can block blood vessels and cause death. The number of clusters, which is seen in figure D, was significantly higher with CLDN 18.2 positive cancers. And in figure E, you can see how in the presence of CAFs, that led to significantly higher mouse mortality rates. It was also found that CAFs can turn on certain genes in cancer cells that help them move and survive. Looking at MMP2, TNC, and ITGA1, which help the cancer cells move and survive, they were expressed at greater rates in the presence of CAFs than with just CLDN 18.2 positive cancer cells alone. And lastly, it was found that a protein called S100A4, which is released by the CAFs, interacts with CLDN 18.2 positive cancers and strengthens their bond that creates the clusters that we discussed earlier. The red and orange bars represent cells with lowered S100A4, and those cells experienced less migration than untreated cells, suggesting that lowering S100A4 and CAFs can reduce how well cancer cells can move.
Interpretation. So the researchers found that a protein called Claudin 18.2 helps cancer cells form strong connections with nearby cells called cancer-associated fibroblasts, or CAFs for short. These CAFs are kind of like the support team for cancer. They help tumors grow, move, and resist treatment. But here's the twist. When cancer cells stick to CAFs using Claudin 18.2, they can travel together through the body in clusters. It's like they're hitching a ride, and that ride might lead to new tumors popping up in other organs. That's what the researchers saw when they put these cells into mice. The tumors grew faster and spread more when the cancer cells were partnered with CAFs and had Claudin 18.2. So what does this mean in real life? It means Claudin 18.2 isn't just a marker that tells us where the cancer is. It's actually doing something. It's helping the cancer cells latch onto these helper cells and form little teams that are more dangerous together than apart. And when these clusters travel through the body, they're much more likely to survive and settle into new places. This is how cancer spreads or metastasizes. That's a big reason why cancers like stomach cancer are so deadly once they advance. And you can see that Claudin 18.2 is the bridge between these two cells. Most cancer research focuses on the cancer cells alone, but this study highlights how cancer teamwork, specifically the relationship between cancer cells and their fibroblast buddies, is a major factor in how aggressive the disease becomes. The researchers also found a potential partner in crime, a protein called S100A4, which is found on the CAFs and may be what helps them stick to Claudin 18.2. Think of S100A4 and Claudin 18.2 like Velcro. When they connect, the cells stay together. And you can see here, when these two different cells are working together, they are a lot more protected and have a higher chance of spreading and surviving in the body, whereas a cancer cell on its own might be caught by the body and eliminated pretty quickly. So did the scientists get it right? Well, From a scientific point of view, the researchers interpreted their data pretty well. They used multiple experiments from cell dishes to mice to support the idea that Claudin 18.2 helps cancer spread by sticking to calves. It all lines up with what they saw. More Claudin 18.2 means more clusters, more growth, and more metastasis. I agree with their conclusions. The only thing that's still a bit unclear is how Claudin 18.2 and S10084 actually bind together. We know two people are dancing, but we don't know exactly who asked who to dance. That's something future research could explore. But overall, I agree with their conclusions, and I think their experiment is very sound.
With all that being said, the study provides valuable insight as to how Claudin drives gastric cancer progression through its interaction with the tumor's microenvironment. Typically, Claudin helps maintain structural integrity by acting as a tight junction. The researchers revealed that within cancer, it becomes a promoter for metastasis through its interactions with CAF. CAF secretes a protein called S100A4, which binds to Claudin, and this interaction helps cancer cells stick together and spread. This new interaction provides a signaling pathway that connects cancer cells to the surrounding molecules. This interaction explains why many patients were not responding well to the Claudin therapy. The CAF creates a new path for survival even when Claudin is blocked. This now gives doctors and researchers new biological information to create new treatments that are personalized for cancer patients. Using this new info, researchers should start testing blockers for S100A4 and other CAF targets, as well as researching more into the interaction between Claudin and S100A4, as well as any other mysterious interactions that may be happening within the cell. But with more efforts toward this issue, I think researchers will be more than set to tackle gastric cancer and possibly learning new ways to handle other cancers in the future.